Hello and good evening, everyone. So today we are gathered to discuss on the lectures of week 9. So as I always do, we will be first discussing the lecture slides in very briefly, and then we would be solving a few questions that would help you in solving your assignments and taking up future exams that would be coming up. Okay, so right, so let us begin with our lecture class, and the questions that we would be solving would be not only helpful for your NPTEL course, but also they are very important for any competitive exam, especially the you say GATE or the NET exams and the different competitive exams that you have like DBT and all. So try to attend these classes, and that would be definitely helping you in the long run. And apart from that, just most people might not find their time in their busy schedule to attend these classes. For them, these lectures are definitely being recorded, so that would help them anytime when they want to see, or definitely for you people who are attending the classes to go back once again and check what has been taught. Okay, so I'm sharing my slide with you all. Kindly confirm, and we will be starting with the lecture. So I believe my screen is visible to you all. Yeah, that's good then. So let us start. So basically in today's class, we would be talking about, broadly speaking, isolation and characterization of DNA, or in general, if I talk, it's about the nucleic acids. Mostly we would be focusing on DNA. So we discussed about in the previous classes, we discussed about the isolation and characterization of proteins. So now we come back to the a more molecular level, that is the nucleic acids, or a more tiny molecules, that is the nucleic acids, and how we can isolate and characterize these small molecules. Cannot call them small molecules, but a combination of small molecules to build our genetic material, or the DNA. So among the biomolecules, we can categorize the biomolecules into four categories. One is carbohydrates, that consists of the sugar molecules, including your glucose, you have the fructose, you have the galactose, and there are other different kinds of um, sugars having 6 carbon, 7 carbons, even 4 or 5 carbons. So these are, the first category is carbohydrates. Next you have lipids and fats. That so in the lipids or fats you have it's a combination of a hydrophilic head and a hydrophobic tail that stays on the inner portion of away from the surrounding water molecules, and it forms the tight core towards the inside. So the hydrophilic head, if you can see, is a combination of phosphates, you have the glycerols, then you can have molecules like choline. Okay, And the hydrophobic tail includes your long carbon saturated as well as unsaturated carbon hydrophobic tails. That so this entire structure builds up to give you, so this entire structure is responsible for forming the fatty acids, or you can call as the lipid molecules or fats. Okay, so if you can see over here, so this is the phospholipid, the PLP phospholipid bilayer, where you have the phosphate group on top, you have lipid molecules in the middle, and that's a sandwich structure. Okay, then you have a few you have the membrane proteins, okay, that are attached to different receptor molecules. You have the antibodies are towards the outside, you have the integral proteins. Okay, so there are different kinds of molecules inside this bilayer structure. These are all categorized under lipids and fats. Next, the most important biomolecule which we have been discussing about since a long time are proteins. So proteins are a combination of amino acids that are responsible for the structural and the functional aspects of our organism. So among these proteins, you have a very interesting category which is enzyme. So enzymes are biological proteins that plays different roles in decreasing the energy barrier and catalyzing the process that has been termed to be slow. Okay, and the last category is nucleic acids, among which you have the DNA, RNA. In DNA, you have the single-stranded DNA. You can have a double-stranded DNA. RNA also, single-stranded RNA. You have double-stranded RNAs that are formed by forming a turn or a loop by forming complementary strands at different sequences. So these are the four categories of biomolecules, starting with the carbohydrates. You have the lipids, fats, proteins, and nucleic acids. Okay. Now, when we speak of nucleic acids, what does it combine or compose of? So nucleic acids are basically composed of sugar moiety. You have the phosphate group attached over here and a base. So base includes your adenine, guanine, thymine, and cytosine for the deoxyribonucleic acids or the DNA, and for RNA or the ribonucleic acid, you have the thymine is exchanged by an uracil group. So this is the entire DNA structure. So DNA can be double-stranded, might be single-stranded. Same works for RNA, single-stranded and double-stranded RNA. So a single-stranded RNA, we are all aware of single-stranded mRNA that is responsible for the translation into proteins, whereas the double-stranded RNA is found by either causing a turn or a loop by forming or getting complementary sequences with the different complementary strands or complementary regions. So in this manner, you also get a few double-stranded RNAs. So nucleic acids, what's the role of nucleic acids? It's known to us. It forms the genetic material of our body. It stores and transmits hereditary information. So DNA stores the information in a genetic code. So these codes are the different amino acids or the nucleotide sequences. And RNA carries information to the protein synthesizing machinery. So there are different types of RNAs that exist. You have the mRNA, you have the tRNA, and you have the rRNA. So the highest number of the highest percentage of RNA is mostly obtained by the ribosomal RNA or the rRNA. Okay, so the mRNA is mostly responsible for carrying the information to the protein synthesizing machinery. So if we speak about the structure of a DNA and an RNA, you have a phosphate group, you have a pentose sugar ring, the ring molecule, and you have a nitrogenous base that varies to give different names to the different uh, bases. Okay, so these bases are named, or this particular structure is named according to the name of the particular nitrogenous base. So we have been studying this since a long time, or we are aware of that, so I'm not going much into details about all of this. So what is mostly important is the phosphate group. Then you have the pentose sugar, you have the 5' phosphate, you have the 3' OH group over here, and you have the nitrogenous base. So this structure is constant. You can see over here, this is the deoxyribose, and you have the ribose sugar. So mostly we are not going into the details of all of that, just comparing the deoxyribose sugar with the ribose sugar. So if you can find the differences between these two sugar molecules, the first observation is the 2' OH. Okay, that is replaced by an hydrogen in deoxyribose, whereas you have 2' OH in RNA. That makes it more susceptible to oxidation, right? And over here this OH is replaced, and you get over here a phosphate group, and this OH is replaced to give a nitrogenous base. So this is the nitrogenous base position, and over here you lose a hydrogen to get a phosphate group. So both deoxyribose and ribose sugar contains 5 sugar carbon sugars, that is the pentose ring structure. Okay, so we discussed about the differences between them. You have it now. Okay, so now talking about the nitrogenous bases, we already know that the different nitrogenous bases includes adenine, cytosine, thymine, uracil, then you have guanine. So there are 5 sugar molecules, nitrogenous bases, and 4 for each, 4 for DNA and 4 for RNA, where only uracil and thymine differs in DNA and RNA respectively. But we need to categorize them according to their structural features. So these nitrogenous bases are differentiated into pyrimidines and purines. So in the category of pyrimidines, you have cytosine, thymine, and uracil. These structures are very, very important for your exams, because there might be questions where you're given a particular structure and you're asked about the name of that particular nitrogenous basis. So it's very important to be aware of these structures whenever you go for any interview or an exam. It is very important to be aware about all of these structures. So in pyrimidines you have cytosine, thymine, and uracil. Just give me a minute. Right. So your pyrimidines include cytosine, thymine, and uracil, whereas your purines contain adenine and guanine. And the other thing is that the pairing. So the pairing occurs between pyrimidines and purines. You have to always remember that it's between the pyrimidines and the purines. So as a unit, these three components, your nitrogenous base, the pentose sugar, and the phosphate um, group forms a monomer unit, and we call them as the nucleotides. So what is a monomer? A monomer is an individual unit that acts as a building block for large biological molecules. They are nearly the identical units, and polymers are large molecules that are made up of the small molecules, like if we think of a chain where we have a short polymer and an unlinked polymer that is joined by the dehydration or removal of water molecule to form a new bond. And whenever we see the removal of a water molecule, only one thing remains we can think of is the peptide chain, okay, that is formed by the dehydration reaction by losing one water molecule to get your polypeptide polypeptides. Okay, so then comes the breaking down of the particular bond uh, with the water molecule. Okay, so that's the hydrolysis of a polymer. So similarly, DNA and RNA are also polymers, where you have, say, thymine and adenine, and you see that there is joining. Okay, so how you get that particular joining? So you have a OH bond over here. So you have a OH over here, and you have an H over here. So that particular thing is lost, and you get that particular chain. Okay, so this is your 5 prime phosphate, and you have a 3 prime OH that will be at the end. Now if you think of a DNA strand, what happens is that you have a thymine, you have adenine, cytosine, and guanine. So this is a DNA strand. Similarly, this is all the joining. This is the 5 prime phosphate group, and as you can observe over here, this is the 3 prime OH, and this is the 5 prime phosphate group, and this is the 3 prime OH one. Okay, right. So if you think of the base pairing, so cytosine base pairs with guanine and adenine base pairs with thymine. So it's basically purines with the pyrimidines and pyrimidines with the purines similarly. So you can see that the number of hydrogen bonds between these two molecules are 3, and between adenine and thymine it is 2. Okay, so 2 hydrogen bonds, and here you have 3, so here you have 3, and here you have 2. So this is the sequence of the nitrogenous bases in the code of DNA, right? So, and if you can see over here, this is 5 prime, and over here you have 3 prime, and during pairing what happens here is the 5 prime, and you have the 3 prime over here. So this is how it twingles. The double helix is formed between the DNA strands, and if you can observe over here that the distance is for the 10 bases is 3.4 nanometer, or 0.34 nanometer is the pitch. So this is the pitch. So this is the pitch, and this is the length for um, 10 base pairs. Okay, so this shows two strands of DNA in the form in which DNA is present within the cell. So this is 10 base pair for 3.4 nanometer, and the pitch of the double helix DNA is 0.34 nanometers. Now talking about the nucleic acid strands, so these are polymers that are derived from the nucleotides. So the backbone consists of repetitive phosphate deoxyribose units, and the individual nucleotides are joined by phosphodiester linkages. Like proteins, nucleic acid chains also have a polarity that is defined by the 5' and the 3' ends of the sequence. So by convention, the sequence of the bases is written from left to right, that is from the 5' to the 3' end. When you are writing the forward strand, so the forward strand is basically directed from the 5' end to the 3' end. That is very simple for the forward strand. So you have a forward strand, you have a reverse strand, where this is the 5' and this is the 3'. So when you are writing for the forward strand, it is very simple. You write it in the same sequence. But when you are writing for the reverse strand, okay, that is from the 3' to 5', you have to also write from the 5' to the 3' direction. So how you write it? You write it the complementary sequence of this particular strand. Okay, so it is also written in the 5' to 3', or you can write it in the 3' to the 5' manner. So the complementary strand of this is this. But convention wise, you write it from the 5' to the 3' end. Like you have over here cytosine, adenine, and guanine, so you write CAG from the 5' to the 3' end. So we see that the cellular DNA exists in the right-handed double helical form. You have the A DNA, B DNA, you have the Z DNA. Okay, but the most common type is the BDNA molecules. So anyway, so cellular DNA consists in a right-handed double helical form. So the double helix consists two interwound antiparallel DNA strands. So the strands are complementary and paired together via the Watson-Crick base pairs ATGC, and the backbones of the strands are located on the outside of the helix while the bases are stacked inside. So as you can see over here, these are the bases that are stacked inside. So if you can see, these are the bases that are stacked inside, and there is a covering of, so this one, the red one, is from the 5' to the 3' end, and the blue one is from the 3' to the 5' end. In between you have the base pairs that forms the minor groove and the major groove. So the minor groove is the one that contains wide spaces. The minor groove is one that are it's a narrow grooves in which the parts of the bases are exposed to the outside. So the DNA binding proteins locate and interact with the specific base sequences exposed in the grooves. So in that you have the major groove, sometimes it interacts with the minor groove, like you have the transcription factors. So the transcription factors are proteins that interacts with the DNA molecules, mostly the major groove of the DNA molecules to carry out different functions, like you have the Hox proteins and all that has different roles in the posterior and the anterior region of your Drosophila and humans. So let's speak of the DNA bending. So DNA can be bent because it lacks the stabilizing bonds. that are oriented parallel to the axis of the double helix. So the bending commonly occurs on binding of transcription factors, that is your Tata box binding protein or TBP, and DNA binding proteins like you have the histones. So because it needs to bind, so it has to twist itself to bind with the different kinds of proteins. Right, so DNA binding is very essential for the packaging of DNA in chromatin inside the eukaryotic cells. Now, just when we speak of the supramolecular structure of different nucleic acids or you have the DNA, so in many cases, what happens is that there are proteins that are of large sizes from 10 to 100 of subunits. So such complexes exhibit the higher level of structural organization known as the supramolecular structures. So your examples includes your mRNA transcription pre-initiation complexes. You have the spliceosomes, you have some proteosomes, then ribosomes, etc. So these higher level of structures functions as macromolecular machines that acts and the different subunits when coordinated in some overall task. Okay, so the protein synthesis by the ribosomes. So these are the general transcription factors that binds the DNA sequences and forms the pre-initiation complex. So this is generally for the RNA polymerase, but other transcription factors and cofactors also binds with the different molecules and carries out the functions. So next, speaking of the base catalyzed hydrolysis of RNA. So RNA is more susceptible, is more prone to this kind of oxidations because of the 2' deoxyribose. Because of the 2' OH group, and the phosphodiester bonds of RNA are susceptible to hydrolysis in basic solution due to the presence of the 2' OH group of ribose. Now, in contrast, the phosphodiester bonds in DNA are much less susceptible because of the 2' deoxyribose lacks this group. Now, it is thought that DNA was selected over RNA as a preferred molecule for long-term storage of genetic information because it is a less reactive molecule due to its lack of the 2' OH group. So this is a very nice natural selection process for storing of the genetic material, because had it not been stored, the genetic material could never have been carried over to the offsprings or the future generations, to the next generation, and the genetic information would have been just lost in one generation. However, it is not the case for mRNA, for RNAs, because they can be received. So the message can be received or transmitted to the RNA uh, by these DNA molecules. So it can be transcribed easily. But once the message is lost in DNA level, then you cannot get it back. Now, speaking of DNA naturation and renaturation, so DNA denaturation refers to the unwinding and separation, that is the melting of two strands of double helical DNA molecule. Like you have helicases that separate the DNA strands. Now, in vitro denaturation can be achieved by heating or treatment with low ionic strength buffers or at extreme pH. So denaturation can be monitored by UV absorption spectroscopy, because the GC base pairs are held together by the 3 hydrogen bonds, and your AT is connected by 2 hydrogen bonds. The higher the GC content, the higher will be the melting point of that particular DNA. Now, in DNA renaturation, double helical molecules are formed by the annealing of single-stranded molecules. So now, speaking of a very interesting term called the DNA supercoiling. So processes such as replication that unwinds double helical DNA introduces some kinds of torsional stress that results in the supercoiling. You observe this mostly in cases of circular DNA molecules like the bacterial plasmids, some viruses, but occurs in linear eukaryotic chromosomes as well. So replication causes overwinding ahead of the strand separation site and the positive supercoiling. Now, underwinding can occur that results in negative supercoiling, called you have enzymes like topoisomerase that regulate the amount of supercoiling in DNA in vivo. So there are two types of topoisomerases. You have topoisomerase 1 that relaxes the supercoiled DNA, and topoisomerase 2 that introduces negative supercoils into the DNA. Now, cellular DNA exists in a slightly negatively supercoiled state, so negative supercoiling facilitates processes such as replication and transcription where DNA is unwound. Now, speaking of the RNA structure, see, you have this different secondary structures, like I was speaking. So, how are these double-stranded RNA molecules formed? So, you have the hairpin structure, you have the loop that is formed, and this is the double helical stem region. You have get also a few tertiary structures like the pseudo knot structure that is formed in this a manner. So you can find the loop region over here. So most cellular RNAs consists of single strand. The higher the double helical regions are common in RNA, where the complementary sequence region occurs. So common types of RNA secondary structure includes your hairpins and the stem loop structures. So stem regions from the A DNA type double helices. So the turns connecting the helices are shorter in hairpins than in stem loops. So RNAs can also form elaborate 3D structures in molecules such as tRNA and rRNA. So the pseudo-knot tertiary structure found in human telomerase RNA, the 3D structures of ribozymes rival those of proteins with respect to complexity. So another important thing, why do we need DNA? So it is firstly from a broader perspective, it is responsible for storing the genetic material of an organism to detect, enumerate the clone genes, enumerate species, detect sequence specific DNA regions, and create new DNA constructs, that is the recombinant DNA that we would be discussing broadly in our next class. And when we speak about RNA, so it is about what type of genes that are being transcribed, when and where will the genes be transcribed, and what is the level of transcription. So this is all about the details of DNA and the nucleic acids. So now coming to why do we need DNA or how can we get DNA. So over here we have DNA purification. So till now, if you have any questions, you can just ask me. If you have any queries, or else I will continue. No questions? That is good. Okay, so let us continue then. So next we will be studying about the purification. So the purification procedure. So what is done? So how do we extract DNA? So let's see. So firstly we grow a culture of bacteria and then we harvest it. So how do we harvest it? By centrifugation. So we get a pellet of cells and we get a cell extract. So the cells are removed and broken to give the cell extract. Okay, so this is the pellet. So we discard the supernatant and we take this pellet and dissolve it, resuspend it, and then we go for DNA purification from the cell extract, and we calculate the DNA concentration. So first is cell growth, then harvesting and lysis, followed by DNA purification and DNA concentration. So thinking of the bacterial genomic DNA preparation of the cell extract, you have to disrupt the cell wall and the cell membrane by lysis using detergents. You can use lysozyme, heat, organic solvents like so some detergents like you have the sodium dodecyl sulfate, some charged detergents, okay, and you have some harsh chemicals like NaOH. So you get the cell extract, so you remove the cell debris and you get the cell extract. Okay, so you have the DNA, RNA and the proteins in this cell extract. So what you do, you collect this cell extract and mix it with phenol. So what happens, you get separate layers by centrifugation because you're mixing it with an organic solvent. So the aqueous layer will contain your DNA and RNA. Your the middle layer will mostly contain the coagulated proteins, and the bottom layer is specifically your phenol. Okay, so this is the phenol-chloroform method. So you need to mix gently to avoid any kind of shearing or breakage of the genomic DNA. You add the enzyme RNase A to degrade the RNA in the aqueous layer. Also, you can use RNase H to to degrade any kind of DNA RNA hybrid molecules, and then you get only specifically the DNA which you can concentrate and use. So the other way of using it is by spooling and ethanol precipitation. So you use 70% of the final concentration of ethanol and your DNA gets precipitated. You collect it by centrifugation and you mix it with water, and and you check the concentration. So by spooling is a glass rod where you have the DNA fibers, you have ethanol and the concentrated DNA solution. So these are the two ways of how you can concentrate the genomic DNA. So the genomic DNA preparation in plants, so how do you get rid of the carbohydrates? So you use detergent molecules in the low ionic conditions. So the precipitated nucleic acid CTAP complex is formed. So in the solution or the supernatant, you have the carbohydrates and the proteins, and in the pellet you have is nucleic acid CTAP complex. So what is this CTAP? It is a positively charged detergent that binds with the negatively charged nucleic acids, and that goes into the pellet region. So you discard the supernatant and resuspend the pellet in 1 molar of NaCl to increase the ionic strength. So here low ionic conditions were used. Now to adjust the ionic conditions, you have to resuspend in 1 molar of NaCl, followed by ethanol precipitation and ribonuclease treatment to get the pure DNA. Now this is all about the genomic DNA extraction from different organisms, plants and the bacterial culture. Another DNA extraction method is the plasmid extraction, that are the vehicles for recombinant DNA. So what is recombinant DNA? We would be talking in the next class. So how do we get the plasmid DNA and isolate it from the genomic DNA? So plasmids are non-chromosomal DNA, replication independent of the chromosome. many copies per cell. It is easy to isolate and also easy to manipulate. That way it's an extremely fantastic vehicle for recombinant DNA. Now, speaking of the plasmid purification, where you have the alkaline lysis, so you have the linear DNA molecule, you use a higher pH using, say, NaOH, that is the alkaline conditions, and you denature the DNA. So, how do you neutralize it now? So, because firstly you have denatured the DNA, so harsh condition is used to denature both the genomic and the plasmid DNA. Now you have to neutralize it or renature it. So neutralization of the genomic DNA cannot occur because they are large molecules and it takes a long time for them to renature. But the plasmids uh, can be renatured because they are more or less small than the genomic parts of the DNA for the particular organism, and they can be easily renatured by a neutralization buffer using magnesium acetate, etc. So there is a tangled mass of linear DNA. So you centrifuge it, the pellet of the linear DNA molecules falls, and you get the supercoiled plasmids. So you extract it and you bind them to the silica column. So what you do, so you separate the linear DNA from the supercoiled DNA plasmids, okay, by the alkaline hydrolysis, alkaline lysis. Then you neutralize it or renature them. Now renaturing them will only renature the plasmid DNA, not the genomic DNA. You centrifuge them at high speed for a long time, you only get the plasmid DNA in the supercoiled plasmids in the supernatant and the pellet at the bottom. You take the supercoiled plasmids and you put them in this silica columns. So the silica columns will bind the DNA well in the presence of high salt and is disrupted by the elution with water. Now you can elute them with water, you can elute them with a kind of buffer known as the elution buffer, where you have some higher concentration of salt molecules for a longer storage. So elution can be done in two conditions. One is water, one is the buffer condition. So the buffer condition mostly contains some salts. So when you have salts in it, that is for the stabilizing the DNA structure and for longer storage. If you're eluting in water, it is for the shorter storage, and you can use it only for a shorter period of time. So what you do, you take the supernatant, you take this supernatant and you load them in this silica columns, and you wait for say 5-10 minutes, and then you elute them with water. So this is your DNA. Whatever DNA is there will bind to this column, the other things gets discarded, and when you're diluting it water, then you get your DNA. You can check the concentration and use them. So for DNA purification, you have the phenol-chloroform extraction as well. So 1:1 ratio of phenol and chloroform is used, or a ratio of phenol, chloroform and isoamyl alcohol in so you have the phenol, chloroform and the isoamyl alcohol. So what does the phenol do? It denatures the proteins, precipitates form at the interface between the aqueous and the organic layer. Chloroform increases the density of the organic layer, and isoamyl alcohol prevents foam. So that is the entire overview that I discussed before. So first you grow the cells, you harvest and lyse them, then you purify them and you check the DNA concentration. You can use them for the recombinant purposes, you can send them for DNA sequencing, and also for different DNA studies. But what is the problem with RNA? We are all aware of it. So RNA is chemically unstable. So spontaneous cleavage of phosphodiester backbone via intramolecular transesterification, because RNA is more susceptible to nearly ubiquitous RNA degrading enzymes or the RNases. So the RNases are released upon cell lysis. The RNases are present on the skin and are very difficult to inactivate because disulfide bridges confer stability and there is no requirement for divalent cations for any kind of activity. That is why RNAs cannot be used. Okay. So we got to know about why the problem with RNA. We were aware of it, but just briefly mentioned it. So now you think of the PCR. Now we will talk about PCR. So what is it? But why do we need to do PCR? For the amplification and different recombinant studies, like you have the cloning, you have to do site-directed mutagenesis, addition and deletion of different genes and all, or playing around with the particular DNA that you have extracted. So this is the DNA that you have extracted. Now you can play around with it. So what is a PCR? So PCR is a polymerase chain reaction. It was first described in 1971 by Klepp and Khorana and redescribed and used in 1985. It allows massive amplification of specific sequences that have defined endpoints. So this is a fast, powerful, adaptable, and simple technique that can be used in many applications. Vastly speaking of engineering, DNA engineering, you can use the genetic engineering followed by DNA amplification. You have cloning, site-directed mutagenesis, and so on. So why do we need to amplify specific sequences? To obtain materials for cloning and sequencing for the in vitro studies, to verify the identity of engineered DNA constructs, to monitor the gene expression, diagnose different genetic diseases, reveal the presence of microorganism, identify an individual, and so on. So there are a lot of applications of these sequences. So what we need for PCR is a template DNA, that is the DNA that we extracted, we purified, that contains the target sequence. So we need primers, we need thermostable DNA polymerase, like we have the TAC polymerase, such that it can survive at high temperatures. We need buffers, DNTPs. Um, we need primers, two types of primers. We need a forward primer and we need a reverse primer. So the primers are short oligonucleotides that defines the ends of the target sequence. So we have the buffer that includes the magnesium ions for carrying out this polymerase chain reaction. And we need a thermal cycler to adjust the temperatures or vary the temperatures and carry out the different cycles. Okay. So how does a PCR work? So there are three steps of PCR. First one is denaturation, then you have the primer annealing and the primer extension by DNA polymerase. So in the first cycle, what happens? It's denatured at a high heat. So this is the reason why we are using a thermostable DNA polymerase to be able to survive at such high temperatures. So, and this stage can also be a single stranded, so that does not matter. So then you denature this particular double stranded DNA or a single stranded, then you don't need to denature it, followed by the primer annealing at temperatures. So if a primer has a melting temperature of say X. So the annealing occurs at a temperature X minus 5 degrees. Okay, so this is what we need to keep in mind. Okay, so first denaturate, then we add the primers and anneal them, followed by the extension using the DNA polymerase. And this cycle is this is the first cycle that has occurred. So this cycle is extended to many reactions, say about 30 cycles, 40 cycles. So this is that's the second cycle. So you can observe that there are many cycles that are occurring. You can repeat it for 30-35 cycles depending on how much you want to amplify the sequence, and you can see the number of DNA strands that you would be getting. Okay, so this is the entire of the PCR reaction. What we need to keep in mind is denaturation, renaturation, primer annealing, and the primer extension. So if you are repeating this PCR cycle for 30 reactions, you see you have 2 to the power, in the first step you have 2 to the power of 0. After cycle 1 you have 2 to the power 1. So after cycle 30 you would be getting 2 to the power 30 molecules. Okay, so that is the level of amplification that is occurring, right? So that is all about the isolation and characterization of nucleic acids, mostly DNA we spoke about, and a few information about the PCR reaction. Probably we would be speaking about the PCR reaction once again, but till now it's a very brief idea. So that is all we had to tell about PCR. And then, so let us now solve the questions and see how interesting the question and answers get. Okay, right. So it's getting recorded. Fine. Okay. So do you have any questions till now? Are you there? Oh, wait. I asked, do you have any questions till now? No questions. Great. Just give me a minute. Okay. Right. So the first question says RNA is chemically less stable than DNA because presence of hydroxyl group, presence of at 3 prime at 2 prime position, presence of ester bond at 3 prime position, uracil well DNA is thymine. Yes, option B is correct because it is susceptible to the uh, alkaline hydrolysis, so it makes it more less stable. So isolation of DNA from plant sources possesses challenges due to the presence of DNA, high amount of protein, high amount of polysaccharide and secondary metabolites, and low amount of DNA. Yes, to C, that is absolutely correct because of the presence of polysaccharides, carbohydrates, molecules, and different secondary metabolites. Fantastic. Those two are the correct options. Going to question number 3, the function of EDTA in cell lysis is to act as a chelating agent, denature proteins, destabilize the cell wall, or all of the above. Okay, you say act as a metal chelating agent. That is absolutely correct. Also, another option is correct, that is destabilizing the cell wall. Okay, right. The next one, what is the function of NaOH in lysis buffer? Denatures genomic and plasmid DNA, degrades the RNA, break down the cell wall, remove proteins from cell lysate. Which is the correct option? So you have said A, denatures genomic and plasmid DNA. That is correct. Any other option seems to be correct? Is there any other option that seems to be correct? Fantastic. Thumbs up. Break down the cell wall. Yes, that is correct. The next question. RNase A degrades RNA that are present in the cell lysate. What will happen if RNase A treatment is not done during DNA isolation? DNA will be degraded, RNA will not be removed as both DNA and absorb at 260. Quantification of pure DNA will be problematic. Both DNA and RNA will form complex and precipitate at the solution, or all of the above. Okay, you are saying it's C. RNase A degrades RNA that are present in cell lysate. What will happen if RNase A treatment is not done? So what is the role of RNA? So RNA will not be removed. That is correct, as both DNA and RNA absorb at 260 nanometer. So quantification will be problematic. This is correct. Both DNA and RNA will form complex and precipitate out of the solution. Just now, so as we are speaking of RNase A, so you see RNase A is responsible for degrading the RNA molecules. RNase H is the one that is responsible for breaking the DNA RNA hybrids. So according to me, option C is not the appropriate answer. Okay, so will form complex and precipitate out of the solution. That probably would have been a correct answer if we are speaking from the perspective of RNase H. But as of now, this one, the quantification would be a problem. So option B is a more appropriate answer than option C. So keep that in mind. You have to see it. If it's only spoken of RNAs, not A or H, then B and C both would be correct. But as we are speaking of RNase A, B is correct. If we would have spoken of RNase H, option C would have been correct. Okay. So just keep this thing in mind. Anyways, good try. What is the role of SDS in lysis buffer for plasmid DNA isolation? Solubilizes the membrane, denatures the protein, disrupts protein DNA complexes, and all of the above. You say it's D? Yes. Denatures the proteins, that is correct. We all know it. Solubilizes the membrane, that is also correct. And disrupts DNA protein complexes. So all of this is correct. So all of the above options are correct. So D is correct. Going to the next one. Isolating plasmid DNA from genomic DNA is based upon. So how do you isolate plasmid DNA from genomic DNA and what basis? Conformation, charge difference, size difference, cellular location. So this is an MSQ. So more than one option can be correct. One option can be correct. Fantastic. So it is based on conformation and size difference. Very good. So elution buffer. I was just speaking of elution buffer. So you elute DNA with elution buffer, sometimes with water. So what does this elution buffer contain? 5 to 10 millimolar tris, pH 8.2, EDTA, water 0.5% SDS. What does it contain? Let us see. Yes, it does contain some salts, like I was speaking. So it contains 5 to 10 millimolar of tris of pH 8.2. Another option needs to be correct. Definitely water is there, so we all know water is there. Yes, water is also there. How can water not be there? The other important option is option B. It is EDTA for chelating the ions. Okay, that can act as the different cofactors. Anyway, so these are the options that are correct. The next one. During extraction of plasmid DNA after the addition of neutralization buffer, white precipitate is formed due to formation of. So in the neutralization or the renaturation step, white precipitation is observed due to what? Insoluble potassium dodecyl sulfate salt, soluble potassium dodecyl sulfate salt, potassium acetate, and none of the above. Yes, because it's forming a precipitate. It's very obvious that it's an insoluble component. So yes, it's insoluble potassium dodecyl sulfate salt. So what happens when neutralizing buffer is added after lysis to the cell lysate? Chromosomal DNA gets tangled and precipitates out. Plasmid DNA anneals and remains in the solution. Proteins and SDS forms insoluble precipitate. What happens? Okay, you're telling D. So yes, chromosomal DNA gets tangled and precipitates out. That is great answer. It anneals and remains in the solution. Fantastic. And proteins and SDS forms insoluble precipitate. That is also correct. So all of the above options are correct. So we are going very fast. That is good. So DNA bound to silica membrane can be eluted by the addition of. So how can you elute the DNA bound to silica membrane? So nucleus free water, trace of pH 6 pH 6, low ionic strength buffer, and 0.5% SDS solution. Yes, nucleus free water, and just now I mentioned, yes, 5 to 10 millimolar of tris solution. So low ionic strength buffer. Fantastic answer. The next one, to wash DNA bound to silica membrane, which of the following reagents are used to wash? So what is used as a washing buffer? Can you tell me what is used as a washing buffer? Is it trace of pH 8, deionized water, ethanol, isopropanol, or detergents? So during plasmid extraction, we have a few important steps where we have firstly RNase treatment, then we have lysis, then we have renaturation. We have lysis by alkaline lysis, then we have the renaturation using the neutralization buffer, then we have the washing step, and finally elution. So what is present in the washing buffer, or what is that washing solution? Any guess? You can just guess it, it's not a problem. Just guess a particular option. Yeah, you say trace pH 8.0, but that is used as an elution buffer. So if you're trying to wash it, it just gets eluted. So pH cannot be used. If I go with the deionized water, that is also used as an elution buffer. Detergents would hamper the DNA. So ethanol or isopropanol, 70%. Ethanol isopropanol is used for washing the DNA bound to silica membrane. Good. So quality of the purified plasmid DNA can be checked. So how can you check the quality of the purified plasmid DNA? Is it size exclusion chromatography, agarose gel electrophoresis, UV spectroscopy, SDS page, that is the SDS polyacrylamide gel electrophoresis. So how can you check the purity of the extracted plasmid DNA? Agarose gel electrophoresis is a very nice method of doing it. Apart from that, UV spectroscopy at 260 nanometer can be used for the detection, where if when we check the ratio of A260 by 280 to check any protein contamination, and 260 by 2 um, so there are two ratios, 260 by 280 and 260 by 220, to see any kind of salts contamination is there or not. So 260 by 280 ratio is very important for checking if there is any protein contamination. So a better ratio of that um, provides us valuable information about the purity of the plasmid DNA. So agarose gel and UV spectroscopy. The next question. In agarose gel electrophoresis, the plasmid DNA runs ahead of the linear DNA. Why? Why does the plasmid DNA run ahead of the linear DNA? Linear DNA is more compact. Plasmids have higher mass charge ratio. Plasmids are supercoiled and migrates faster. Yes, that is the correct option. They are supercoiled and they migrate faster. So the last question. Neutralization buffer contains. What does the neutralization buffer contain? Sodium chloride, potassium acetate, and calcium acetate. I just mentioned it in one of my. Oh, we gave an answer, right? Oh, let's see. Okay, fantastic. Potassium acetate is the correct option over here. Yeah. So we are done with the questions. I guess there were a few interesting questions for you, and it was a lot more uh, information. There were a lot of information that you got. So do you have any questions? If not, okay, then thank you. We will again meet in the next week and talk about a new lecture. And also these lectures are being recorded and they will be posted in YouTube, so you can anytime come back and look at them whenever you face any kinds of difficulties. Also the slides are shared, so there is not a big deal, so you can check back with the answers anytime you have a problem, or you can also write back to me in my email, or even in the comment section of YouTube. So looking forward to a better understanding. Okay, so thank you all for joining, and we will again meet in the next week. Thank you.